Hi, my name is Rocky Ng, and today I'll be talking about the human neutrophils killing the parasite Trichomonas vaginalis. Trichomoniasis is the most common non-viral STD in the world. According to the World Health Organization, 10% of women of low income are affected by the disease. Many patients do not develop symptoms. However, women who develop clinical symptoms like the strawberry cervix, as shown on the right, compared to the normal cervix on the left, other symptoms can include itching of the genitals and foul vaginal discharge. This neglected disease is caused by the extracellular parasite Trichomonas vaginalis. As an extracellular parasite, we know attachment and cytolysis to the host epithelial cells are important stages to establish infection. However, the immune response to Trichomonas is poorly understood. We are interested to study how neutrophils respond to trichomonas during this infection. To begin this experiment, we first put neutrophils and trichomonas together in the same culture. To put this in an analogy, we are basically observing the parasite and our immune cells in a boxing ring. Who will win this fight? It is known that neutrophils are able to kill foreign cells using three mechanisms. Number one, NETosis. The neutrophil spills out a network of DNA to capture the pathogen. Number two, phagocytosis, which means whole cell engulfment to the pathogen. And three, degranulation, which means spitting out toxic granules to combat the pathogen. What we found is that when you put the parasite and neutrophils together, neutrophils do not kill the parasite Trichomonas by netosis or degranulation. Instead, it uses a similar yet different process than phagocytosis, and this process is called trogocytosis. Trogo means to nibble. We found that the neutrophils are able to take bites off the parasite instead of whole cell engulfment. As shown in the video, the green labeled parasite cell membranes are bitten off by the neutrophils. So what's next? We still don't know what are the molecular player to trogocytosis. Further studies will need to examine other cells using trogocytosis and identify potential genes mediating the trogocytosis process. A potential experiment will be doing a transcriptomic analysis with neutrophil exposed by the parasite and see the transcriptional changes using bioinformatics. So why do all these matter? Since Trichomonas vaginalis is one of the most common STD in the world and most infections are asymptomatic, there are reasons why studying trogocytosis is important. Understanding the molecular players in trogocytosis will help study other cells using this conserved cell killing process. More importantly, by studying the immune response to this parasite will potentially develop alternative treatments to this neglected sexually transmitted disease.